Hi. First, I would like to introduce myself. I'm Louriki Sara, a PhD student at Ibn Tufail University and ICARDA. My talk today will be about the evaluation of seedling resistance to Septoria trittici blotch in diverse subsets of wheat. Today, wheat is grown on more land area than any other commercial crop and continues to be the most important food grain source for humans. It belongs to the grass family, along with barley, maize, oat, and rice. On average, for the period 2014 and 2018, Morocco produced 6.1 million tons of wheat grain. In 2019, wheat production is expected to decrease from 7.3 million tons gathered last year to 4.9 million tons, a decline of almost 20% compared to the average. During the long growing season, wheat is exposed to various fungal diseases that can cause significant yield losses. Septoria trittici blotch, caused by the ascomycete Zymo-Septoria trittici, is a major disease of wheat in all growing areas of the world when climatic conditions are favorable. The infection by this pathogen is initiated by airborne ascospores and dispersed conidia produced on residues of the previous season crop. These ascospores entered into the leaves via stomata and cause primary infection on seedling leaves. Secondary infection of Septoria trittici blotch is initiated by conidia, which form readily in high humidity, particularly if there is water present on the leaves. These spores disperse through rainwash and splashing, causing the spread of the disease to uninfected leaves. Thus, many cycles of sexual and asexual reproduction during the growing season allow epidemic to develop rapidly. Host plant resistance and the use of fungicides are two of the main approaches used to limit yield losses due to Septoria trittici blotch. However, the use of fungicides is not environmental friendly, and their effectiveness has been constrained due to the evolution of insensitivity in pathogen population. Thus, breeding for disease resistance remained the most economic, effective, and environmentally friendly strategy to combat Septoria trittici blotch. In order to breed resistant cultivar and reduce yield losses caused by Septoria trittici blotch, it is urgent to identify highly resistant germplasm in different wheat subsets. For this, in this study, we used 3 different sets of wheat. LPM panel containing 377 accessions, figs subset, and wild relative containing 105 and 35 accessions respectively. For each accession, 5 to 4 seeds in tree replication were sown in a tray of 98 cone, supplemented with NPK fertilizer in a grow chamber with 60 hours light and 8 hours dark. 2-week-old seedlings were inoculated with 100 ml of spore suspensions, and after spraying, the seedlings were covered with plastic bag for 72 hours to maintain humidity and enhance spore germination and infection process. The bags were then removed and the plants were kept in the greenhouse under the same conditions. And after 21 days post-inoculation, wheat responses were evaluated by visual estimation using 0 to 5 scale, where 0 is highly resistant and 5 highly susceptible. As results, among 517 wheat genotypes tested, 70 genotypes were found to be resistant and highly resistant. In contrast, 115 genotypes were susceptible. These results demonstrate that we have a great variation in response of wheat genotype to the Moroccan isolates and that wheat germplasm has diverse sources of resistance to Septoria trittici blotch. As a future endeavor, wheat accessions will be genotyped in order to identify linked markers to resistant loci. As a conclusion, These new sources could be excellent potential candidates to be employed in wheat resistance breeding program in order to develop new varieties with an effective and durable resistance to Zymoseptaria triditi. Thank you for your attention.